Retina Rounds, podcast number 30. Retina Rounds is excited to welcome a true leader in ophthalmology and a giant in retina, Dr. Jay Duker. Dr. Duker is one of the most accomplished figures in our field, having served as Chair of Ophthalmology at Tufts Medical Center, where he helped shape both clinical care and research for over 2 decades. He is now President and CEO of EyePoint Pharmaceuticals, and in today's episode we discuss his remarkable career journey, his transition from academia to biotech, and a fascinating deep dive into tyrosine kinase inhibitors, clinical trial development, and what it really takes to bring a new therapy from concept to regulatory approval. Let's check it out.
Well, it's great to be back on the Retina Rounds podcast. Today we have a special guest and longtime friend, Jay Duker. And Jay's journey in retina has been meteoric and illustrative, and he's definitely one of the luminaries in our field. And Jay, it's really a pleasure to have you here on Retina Rounds. Thanks for joining us.
Oh, it's my pleasure, Steve. Thanks for inviting me.
What we've done with our Giants in Retina series is started off with questions about your journey. How did you come into medicine and then ophthalmology and then retina?
Yeah, so that's, again, it goes way back.
Can you remember?
I think I decided when I was probably 5 years old that I was going to be a doctor. And my mother, in her sarcastic self, said, "You can't be a doctor. They wash their hands 20 times a day. You never wash your hands." So, I don't know how that all fits into the story. But yeah, I was always interested in science. And I went to college thinking I was going to be a theoretical physicist.
Where did you grow up?
I grew up in Pittsburgh.
Uh-huh. And where did you go to college?
Went to Harvard.
Uh-huh.
And thought I was going to be a theoretical physicist until I realized that theoretical physics required a huge math background, which I didn't have. And I kind of liked talking to people. And so I thought, again, medicine was probably a good field for me. So I switched and did the pre-med requirements, went to medical school in Philadelphia at Jefferson, which is now called Sidney Kimmel. And that was really, a lot of things happen in life, and you call it serendipity. Maybe it is, maybe it isn't. But it just so happens that Jefferson was affiliated with Wills Eye Hospital. And that was a huge influence on me going into ophthalmology. And I remember probably as a first-year medical student, when we did the anatomy of the eye, I thought, oh my God, this is fascinating. And then decided I would try to do a little bit of research over at Wills Eye, and ended up working with the pathology group over there, and writing a paper or two, and ended up doing my residency and fellowship there. But it was really strongly influenced by the fact that my medical school just happened to be affiliated with one of the best ophthalmologic institutions in the world.
Yeah, an amazing institution who's churned out incredible leaders in our field, both clinically and academically. And how did you decide to go into academics?
Well, so I was really excited about answering basic questions. Why did a person respond to laser and another person with the same disease didn't? And my mentors at Wills, almost all of them in the retina group, were really academically oriented.
Who were
That was
they?
one of the Gary Brown, Bill Benson, Bill Tasman, Jay Fetterman, Bill Ansley, David Fisher. I don't want to leave anyone out.
No,
But they were all terrific. They were all and great mentors and really, really good surgeons. And that's the other thing you don't realize. When you train at a certain institution, you don't really know what the culture is necessarily elsewhere. You don't know how good the quality of care is elsewhere till you go elsewhere. And you realize, boy, that was really a unique institution. And the reason it was unique, which, some people may say is not a good thing, is it was essentially a private practice that was an academic institution. And as a result, the academic hierarchy wasn't this pyramid with the chair at the top. They were all pretty much equals. And you earned your status by publishing papers, not by necessarily putting other people, behind you, let's say. So it was, I believe, a unique academic institution, and that carried over when I went out into practice. Yeah, it still is.
Yeah.
And when I eventually became chair of an institution, which was full-time academic, I largely modeled it after what I had grown up with at Wills Eye,
Which is
meaning
one of the
we were
things
what
I
I,
admired so much about your institution, by the way,
yeah.
hierarchical hiring, but everybody had agency once they were in.
And you know what? When I'd introduce my colleagues, I wouldn't say they work for me. I'd say we were colleagues because that's what I truly believed. I really didn't want there to be a hierarchy. And the other aspect of it is, as you probably know, being an administrator in some parts of your life too, a lot of physicians really just want to do their thing. Meaning, whether it's see patients, it's operate, it's write papers, it's run their lab, they want someone else to largely take care of it and make it the trains run on time and whatever. And so most of the people in my institution were very happy as long as the trains ran on time and we had an open discussion on things like overhead and how you might get recompensed for doing things like academics. We had all those open discussions. And so it really became a place where I think it becomes self-selecting. The people who like to work under those circumstances flourished.
When you were
But it was
coming out
really
of fellowship, how did you decide that you were going into academia? It was because of your
I
curiosity?
did I didn't. My first job was private practice. And so,
It's
you know,
fascinating because that makes
yeah,
you
well,
in a
well, yeah.
small group.
There's a story behind it too. I married and am still married to my wife, Julie, over 43 years now. And she's a
Which makes you,
Bostonian.
by the way, one of the luckiest guys on planet Earth, but we'll talk about
Thank
that
you.
off the podcast.
Thank you. Yeah. She's a Bostonian, and I love Boston. I loved it, but when it came time to finding a job, that's where I primarily looked. In retrospect, I have to say I can't find much negative about how my career has unfolded. But I would have loved to stay at Wills. I I could have stayed. I'd love to stay, but my wife wanted to move back to New England, so we did, and I joined the Retina Associates, Dr. Scapins' group. And I was the 13th retina specialist in that group in 1990. Now, if you think about that, we have a lot of big retina groups now. This was 35 years ago. There was no other big retina group like it anywhere. It was the biggest in the world. And what I didn't know at the time was, Dorothy, you're not in Kansas anymore. Jay, you're not at Wills Eye anymore. It was a pyramid hierarchical, and not to take anything away from the accomplishments of the people in that group. It wasn't what I was used to. And I did manage to do a little research while I was there and get some things going, including I started to be interested in drug delivery to the eye uh, because the Ganciclovir implant was in development at the University of Kentucky, but a colleague there in Boston was involved with it. It got me involved, and that's one of the reasons I started to become interested in drug delivery to the eye. But anyway, what I didn't know at the time was there was the group was breaking up, and coincidentally, Tufts decided that they wanted to develop their own department of ophthalmology. They had a small one for years and years. It was like three or four full-time people. I don't think they had a YAG laser at the time. And they just decided they were going to get their own department of ophthalmology, really build it. And Carmen Puliafito was the first new chair there. And he recruited me at the time to become the head of retina. And I did. Uh, there were four of us. It was Carmen and me and Joel Schumann, who's at Wills now, and Mike Raisman, who's in private practice. They all came over from Mass Sinai and uh, built a department. And, Carmen and I owe a lot to Carmen. He really obviously assisted me in my imaging research. Just again, luckily, that was where OCT got developed, and that enabled me to really take an interest in imaging. Uh, but Carmen was a guy who he thought at 30,000 feet. He was not an operational guy. And that's what I was good at. So Carmen would have some idea about doing something, and I was the one who was always responsible for carrying it out. Uh, and the relationship, I have to say, worked pretty well. We really expanded the department. And then, as you know, and some of your listeners may know, Carmen left and went to Bascom Palmer to be chair there. And I then was elevated to be chair of the department.
What year did you become chair?
2001.
Wow.
Uh, yes. And remained chair there for 21 years. I stepped down in 2022. Uh,
Let's talk a
and
little bit. Let's talk a little bit about your tenure as chair because you grew a great department. But what I'm really interested in, and I think our listeners want to know, is you put out so many leaders in the field out of your department, whether in
yeah,
fellowship, trained a lot of our residents, turned out to be great docs, trained a lot of great fellows, a lot of great residents, a lot of leaders across ophthalmology. When residents came to you and said, I'm struggling with what specialty to choose, but almost more importantly, I'm struggling with whether to target academia or target private practice. Private equity wasn't really a thing so
no,
much when you were there,
yeah,
but
yep.
how did you advise them, like on a personal level? Because you're such a warm, caring, empathetic guy. I know that you gave them the time of day and more. So what was that conversation like?
So the specialty discussion was usually pretty easy but I would say, well, what brings you joy? Do you like to do the same surgery over and over and over again? Or do you like to be challenged? And, that's one of the reasons I picked retina is that, I use the Mike Tyson quote all the time. Everybody has a plan till you get punched in the mouth. And the plan in retina was, okay, I'm going to go in and peel this membrane, et cetera, et cetera. But what if something goes amiss? And, there are most cases, I won't say it goes amiss, but it's not how you planned it. And that's what I loved about retina. But we had residents who said, I just want to do the same thing every time. Or they were very meticulous. They'd go into cornea. Or, they wanted long-term care of their patients. They wanted that care of the chronic patient, and that's glaucoma. Or they didn't want to ever look at a slit lamp again, and they went into plastics because they had that side of the brain. So most of the time, it was pretty easy to figure out by talking to them what they wanted to do. And the other big part of it, it's really hard sometimes to separate when we've spent all our lives in thinking about the next step. Think about it, dude. High school, I want to be a doctor. I got to get into a good college, get into a good college. The rat race we go on, and it's always the next goal. Then all of a sudden, you're done. You're done with fellowship. Well, now what? Well, now it's completely different. No one's testing you on your knowledge anymore on a piece of paper or an oral exam. You got the patient in front of you. And so that's the other thing that the residents really had to start to adjust to, which it wasn't about the next step. You're there, you're going to be there soon. You're going to be an attending and seeing patients. So what brings you joy? Do you just want to see patients, which is perfectly fine? It's a great career. We have a great career. But do you want to ask questions or do you want to have some type of hybrid situation where you can ask the questions and try to answer them? And so that whole argument about that line between academics and private practice, it was very clear back in, the 1990s. But what obviously has happened to retina is most of the publications and let's say academic work that's not basic science, bench science is coming out of the private practices.
Well, you and I had a lot to do with shifting paradoxically back in this is like submacular surgery trials and
Yeah,
back in those days we shifted or took part as academics shifting
sure.
clinical trials into the community
Yeah.
and we created a monster. I mean, it's a beast now
Well,
and you know better than anyone from your vantage point
yeah,
now.
I
It's
don't
really
know.
hard to do clinical trials at academic centers now.
It is in that it doesn't seem to be getting any easier. And of
No,
course,
that's
I'm
a
on the other end
euphemism.
of it now
Right.
where, my friends can say, hey, I'd love to do your DME trial. And the problem is look, by the time you get it through your RB, we're fully recruited.
Right.
And
It's a different world. But let's go
yeah,
back to this conversation about what specialty to go into and then
yeah,
academics and private
yeah.
practice, because the second half of your tenure as chair, there was this hybrid. I want to be in private practice, but I also want to
Hmm.
do clinical research. How
Yeah.
did that factor in? Because that's a lot of the answer. I want to do both. A lot of
Well,
our trainees are have your cake and eat it too.
but remember the other thing is that there's more to go into choosing a practice than just that decision.
This is what I want to hear your opinion on.
Well, the number one thing is it's the old cliche about it's like getting married. If you go into the meeting with your potential employers and you don't get that warm, fuzzy feeling, it isn't going to get better once you sign on the dotted line. This is the best that it's going to get. And if you see how they practice and you don't agree with it, don't be foolish to think that you, as the one coming right out of fellowship, knows everything, and you're going to tell these people who've been in practice for years that they're not doing it the way they should be doing it.
Man,
It
have I
isn't going to
seen
work.
that mistake happen a few
It
times
isn't going to
recently.
work like
Yeah,
that. And so the other thing is do you want to be a decision maker or not? Once you join a group that's more than, let's say, five, six, seven people, the odds of you as a new employee, eventually new partner, that's going to be a decision maker in that practice, it's decades down the line. So if you're perfectly content with the way the practice is run and you like the culture and you like the places you're going and everything is great, you don't have to be involved in that. But if you're a priori, you're coming out of fellowship going, I've got some skill set in operational stuff and I want to be heard, don't join a 20-person practice. It's just there's no room. It's just not going to happen right away. The
right.
other warning I would give people is not of completely avoid someone in solo practice. But I used to say people that are in solo practice for a reason. And if they've been in solo practice for a long time, you are not going to change them. You have to accept that this is the way it's going to get done until they retire. And there's, again, nothing wrong with that. But you can't go into joining a single person who's been in practice for 25 years and you're going to change her mind about which EMR to use or how to operate. People aren't like that.
Right.
So
George Williams and I used to say all the time that it's really hard to change culture.
yes, yes and no. I'm going to say when you're dealing with a culture of a group that's 20 and you're not coming in as the CEO or the president, yes, you're not going to change that culture. It's a little different in business when you come in a CEO position. That's a little different. But the other thing is, and I mentioned this in my decision, if you have a spouse or a significant other, this better be a team decision because if they're not happy, you're not going to be happy, or you are going to sacrifice your relationship for your career.
Right. I
And
always used to tell them if it's
yeah,
truly a significant other, then treat them that way. If it's an
yep,
insignificant other, then you can make
yep,
whatever decision you want.
yeah. And I used to interview fellows with Trex Topping. You may know that we had a combined fellowship with
Absolutely.
ophthalmic
I
consultants of
sent you a bunch
Boston,
of fellows.
which was unique because it was private practice and academics. You got the best of both worlds. It was terrific for everybody. And yes, by the
Some of
way,
our some
we,
of our
we
favorite
love
residents went to that fellowship, like Darren.
and not to take away from any other residency,
Right.
but Stein, we had a great run with Stein residents. We really
Right.
did. They were
Whole line of
terrific.
them, Reed,
Whole
Gold,
line of them.
all of them.
Oh
Yeah.
yeah, yeah. Anyway, what Trex used to say, and I think this is good advice to the fellows who were coming in for interviews, is have a 5-year plan with your significant other. And after being in a place for 5 years, sit down with them and say, is this what we want? Are we happy here? And if the answer is yes, keep going. And if the answer is no, look somewhere else. Now, I think, Steve, our generation, and I think you and I are aligned with this. How many jobs have you had now in 30 odd years? Two, right?
Yep.
Until I joined EyePoint, I had two jobs, a private practice and an academic job. But I do think this next generation is more attuned to switching. They're not necessarily looking at their first job as their last job. And that's another thing that I think has really changed generationally. I think most of us, when we came out of fellowship, we looked at it as this is a really important decision, and I hope to join a place that I never want to leave.
So when you were chairman, I want to know, besides the academic challenges and the organizational challenges at the university, speak about for a moment what your biggest accomplishments in terms of everybody knows you made giant contributions to drug delivery and OCT, but for you personally, what gave you the most happiness, most joy when it came to what you actually brought forward into our field? Because you made a lot of contributions, more time
Yeah.
than I have to list right here. And then
So
we'll use that to transition to how you decided to move on.
yeah, so the best things were really, and I think a lot of people in academics will agree, it isn't the papers you published, it's the trainees that you trained. And what was so gratifying is, sometimes I get phone calls from a group that one of our fellows had joined. and they would say, "Thank you. I can't believe how well trained this fellow is." Or they've come and shown us things that we never really thought about. We had a fellow join a very large group in the Midwest who, within 6 months, became the go-to person for suturing IOLs. And so that's the kind of thing where, like, you come back and say, you know what? I think we're doing this right. I think that we have a really good training program. And watching the learning curve from that first time in the OR till, for some people, it only took 3 months. Some people, it took the full 2 years to really be comfortable behind the microscope with the decision making, because it was really all about decision making. We talk about hands all the time. Is it really hands? It's decision making. In knowing when enough is enough and understanding that what you're trying to accomplish. So that's the number one thing. I'd say the number two thing was the lack of attrition in my department. Most people stayed a long time, which told me if they had that Trex topping 5-year plan going, they were pretty happy with the way things were going. Because what I tried to do, and I think this is an easy mantra to remember, you hire great people and you keep them happy. And what keeps one person happy can be completely different than another. And you have to actually understand individually what they're looking for. And again, most of the time, what keeps doctors happy is a great place to practice with minimal headaches and an openness so they understand what's expected of them and how they're eventually going to get paid for doing the right thing. So that part of it, in a sense, was easy.
Fantastic. So here you are, you're 20 years into your chairmanship, 30 years into your 20 years into your chairmanship. You survived COVID, you've had a foot on boards and done a lot of very high level consulting, and you've run this great department. You've trained all these leaders in our field. You've made major contributions. You've got a really happy department. You got great people across every subspecialty, including retina, but great people across everything. A lot of them have made big industry contributions. I think about, Goldstein and others, and they're just
Mhm.
remarkable. And
Yeah.
then all of a sudden, you decided did the light go on, or was this like a creeping
No,
decision?
it wasn't all of a sudden. So there were a couple of factors here. And one of them was colleagues and I at Tufts started a company years ago that was a gene therapy company for dry AMD. And we took it from laboratory to through 2 phase 1 trials, and it
I
eventually
remember. What was the name of that company? It was good.
Mera Biosciences.
Yeah.
And it eventually
In
exited
fact,
to
the scientist, I forget his name, he worked with us at UCLA.
Rajendra Kumar Singh.
Yeah,
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 brilliant guy.
yeah.
But we eventually exited to Johnson and Johnson. It's currently in a phase 2, going to read out soon. But that really was exciting. That got me
And
the
that was a
bug.
complement inhibitor gene
It
therapy,
is
chronic protein delivery.
gene therapy, single injection, making CD59, which is a natural
Right.
inhibitor of complement. Anyway, right towards the end, let's say, I don't know, probably I think it was maybe 2018. The administration at Tufts came to me and said, Jay, we're moving on from our dermatology chair, and there's nobody in the department who can take
I
over.
remember this.
You can run outpatient departments. Would you be the interim chair of derm?
I
And I
remember
said,
this.
sure. Why not? I've always told them I'd help. So
Yeah.
I became the interim chair of derm. And you know what was easy? There's some of this was just really easy, I have to say. The derm department was in trouble. And I'll give you an anecdote. So the first day I become chair, I said to the administrator, walk me through the patient experience. So we will follow a patient through the whole thing. And it was going pretty well. And at the end, the administrator says, Dr. So-and-so wants to see you in 3 months, but our schedule doesn't go out that far. Give me your phone number and I'll call you. And I went, what? Got them all together. And I said, you let people leave without an appointment? They're never going to come back. They want an appointment. Well, we don't know in 3 months how many patients the doctor's going to see. And they said to me, it all depends. Dr. So and so will see 20 patients if he has 3 residents. If he has 2 residents, he'll see 15. If he has 1 resident, he'll see 10. And I said, I'm the chair of ophthalmology and I'll see 50 patients, whether I have 1 resident, 2 residents, or 0 residents. This isn't going to last. And it turned out the residents were the ones who actually made the schedule. So these attendings were just kind of do
Laissez-faire,
whatever
right?
you want to do. So we cleaned that up, and I was able to hire, I think, 5 full-time people when I was interim. We fully matched the thing, and then hired a great person. But then I was out of a job, and I had taken a day and a half out of my schedule to do this. And I started to think, like, what am I going to fill my time doing? I've been there, done that in a lot of ways. And, my department, I could continue till I'm well into my 70s and but that's not fair because transitions are hard. And 21 years at the helm might have been too long, to tell you the truth, because change, you can't fix everything. Change needs to be done. So that's when I got interested in actually saying, I said to myself and my wife, if I'm ever going to be a CEO in a biotech company, this is my last chance. So I did.
And let's hear about the company. What was the opportunity like? I mean, you had a lot of choices, and this is a
Yes.
great one that you chose. And
Yeah,
so,
yes and no. This turned out to be great first of all, for some simple reasons. I knew the company really well. The company originally was called Control Delivery Systems. It produced the Ganciclovir implant, which I was one of the first to, Dan Martin and I, worked on that years ago. And he and I, probably each put in about 300 of those back when we saw CMV retinitis. So I knew the
yeah,
technology.
Barry and I spoke about CDS extensively.
Yeah.
Barry Cooper and
And then
I.
it became Cyvita.
Right.
And then it became EyePoint, same company, new technology. Uh, if the company had been in Los Angeles, I probably wouldn't have done it. But
You would look good in a Laker hat. I've told you this over and over again. But anyway, let's Dodger hat, Laker hat, whatever it is.
yeah, that was this was part of it. And I knew the company. I joined the company on the board in 2016. And then the company was shifting from being a commercial company selling YUTIQ in another product to being a pipeline biotech company with DuraView. And I had the opportunity to essentially design the phase one trial and
Which is
design the
awesome.
phase
Can
two
I just
trial.
stop you for a second?
Yeah.
Because I need you to explain to me what DuraView is.
Okay.
What's the science? What's the indication? Like we've
Yeah.
never heard it before. Sorry to get the
Fair
CEO to
enough.
do this, but make
No,
it.
no, this is good. I was, I should have done this from the start.
No,
And
I really,
yeah.
really, I want to know
So so
why. And then at the end, I'm going to ask you, why would I ever use it? So,
so DuraView is a small molecule tyrosine kinase inhibitor. TKIs are been around for a while, and they're big in cancer, approved in cancer, and we've tried them in the eye before, topically and orally
and
for
what do
wet
they
AMD.
do?
They block the VEGF receptor. All the approved products are biologics that block the ligand extracellularly. TKIs cross the membrane, get to the receptor, and block the downstream receptor. And it blocks all VEGF,
So it
blocks
blocks the
PDGF.
receptor from coming out onto the cell surface, or it changes the
Oh, no, no,
conformation
no,
of
this
it.
is intracellular. This blocks the downstream effect
So
when
once
VEGF,
the ligand attaches to the receptor sends a signal, and the TKA
nothing.
blocks
We
the
cut the
signal.
signal.
Right.
Yep. Cut the telephone.
And it's a small molecule.
It is. So a
And
small molecule, the problem is you got to, if you've injected it intravitreally, it'd be gone in a day or two. You can take it orally, and our drug vorolanib was tested orally by a company called Tyrogenex. It was called X82 at the time, and it had pretty good clinical results,
I
but it
remember
had
that.
systemic toxicity. So
Right. Off-target effects. So how do
yeah,
you deliver it now? What sets it apart? What makes it stick around and work?
well,
Why are you
that's
excited?
the beauty of Duracert, which is the name of our sustained release technology. So essentially, you take the drug, the API, and you mix it with a matrix, and then you extrude it into long lengths of drug, and then you chop it into little pieces and you load it into an injector, and it's injected into the eye like any other intravitreal injection, but it's a insert, not a liquid. And the insert is fully bioerodible. What's interesting about our insert, we've used inserts like Ozurdex, we've had them for years, but our insert's 94% drug. It's only 6% matrix. So we've managed to really
What's the
increase the drug.
matrix made out of?
Oh, we don't really talk about that publicly.
Sorry.
It's proprietary. No, it's happy
Good
dancing.
stuff. Well, it's made out of
Yeah,
good stuff that is
it's the
non-toxic
secret sauce,
and it
and
goes away completely.
it should go away completely. Yes.
So
The,
over
the,
how long does one dose last?
Okay, so that's a good question. So the dose, and this is based on animal data, because PK in human, we're not tapping vitreous or taking pieces of the cord to figure it out. So you can extrapolate by looking at PK in the serum. We actually have a very sensitive assay for vorolanib down to the picogram per ml, which is like 1,000 times less than the IC50. We can measure it systemically, and you can look at animals and you can get a ratio between the animal and the human eye. That's a pretty good estimate. And so based on that and based on some of the PK data we have, the drug is basically gone from the initial injection in 9 months.
Wow.
It's variable though. So
When does it
virtually
drop below a significant activity level?
8 months, 8 to 9, but it's
Wow,
variable. And
congratulations.
so it pretty much 100% of humans, we expect for 6 months it to be well in the range of activity. Now, it not only blocks VEGF, though, this is important. Vorolanib is unique. It blocks PDGF because the VEGF and PDGF receptors are very similar. So most of the TKIs that block VEGF will block PDGF.
So explain to our audience the importance of platelet-derived growth factor and the fact that it's
Well,
part of the VEGF family.
I'm gonna just put it out there.
Placenta, sorry.
It's theoretical.
Yeah.
PDGF is responsible for fibrosis.
Mhm.
So in the long term, if you block PDGF, you should block fibrosis in these CNVs.
Right.
We don't know that for sure yet. We will be studying that. We're in pivotal trials right now.
My understanding is that it may have also supported fibrosis, not only caused it, but you might even see some regression of fibrosis. Have you seen any of that?
Again, it's certainly conceivable, but until we can show it clinically, I'm not going to go out on a limb and say, oh, yeah, we're going to be antifibrotic. And so your patients are going to see
So
better.
can I ask you, is this a public company?
Yeah.
And so everything you're saying will put at the beginning the disclaimer, if you like.
Yes. Forward-looking statements. Yeah.
Yes,
So
we can do that.
EYPT on NASDAQ.
Yeah. Okay.
Yeah, we've been
And
public
the
for a
name
while.
of the company is EyePoint.
EyePoint.
The name of the drug is
It's vorolanib in the intravitreal insert. It is called Duravyu.
and Duravyu is in phase 3 clinical
Wet
trials.
AMD, we have completed enrollment in 2 phase 3 trials.
And when do they read out? More or
The
less.
first one will read out the middle of next year, 2026.
1 year
Let's
time
call
point
it-
from last patient in.
Yeah, 56 weeks. Summer, it'll read out. The second trial, the Lucia trial, will read out about 2 months after that.
And are they both domestic or are they both global?
The first trial is 100% US. Second trial is 80% US. The rest,
And
Europe, Brazil, India.
What other anti-VEGF agents are the patients getting, if any?
So the way the trials are set up is that everybody gets loaded. So on day one at randomization, everybody in the trial gets Eylea, 2 milligram, and they get it at week 4, they get it at week 8. And then at week 8, if they're randomized to our arm, they get 2.7 milligrams of Duravyu. And if they're randomized to the control Eylea on label 2 milligram arm, they get a sham. Then they're seen monthly. In the Eylea arm, every 2 months, they get another Eylea. They get a sham to masking in our arm. And then 6 months after the initial dose, so around month 8, on that visit, they get another Duravyu and no sham necessary because that's an Eylea visit for the control arm too. Now, there is an allowance for what we call supplemental treatment. And so for all of
So you don't
these
use the term rescue, you use supplement.
I
like supplement.
You can use either. It's not truly rescue, though. It really isn't. Because in the real world, we won't think of it that way. But I'll get to real world usage in a second. But the point is, if an eye has new fluid with decreased vision from best on study, and it reaches a certain point, more than 5 letters and 75 microns of fluid, then the investigator is required to supplement. And that's either arm. So what's interesting is we did a very large phase 2 trial called the DAVIO 2 trial, which looked at similar kind of structure, but didn't have reinjection at month 8. And we allowed supplements in the Eylea arm.
Of course you
So
did.
on a day you're getting a mandated Eylea shot that you can't be supplemented, but 6% of the Eylea arm by month 8 got a supplement on the off month. Now
So they're
that
getting
shouldn't
seen
surprise
monthly.
you.
They're getting seen monthly.
Because
Doesn't
they were eyes
surprise
that
me at all. It
probably in the real
surprises me
world.
that it's that low.
would have gotten monthly. Well, they had to meet requirements. Remember,
Right.
you might say, hey, in my office, I'm not going to wait for 5 letters and 75 microns.
I'm
But
a
these are
programmed
eyes that
retreater. I'm one of the
these
last dinosaurs.
yeah, these eyes were that met criteria on the off month. And at 1 year, it was 13%. Now, the other way of looking at it is what's interesting is if you look at the Eylea arm and you ask the question, forget about whether you call an injection a supplement or a mandated injection. How many of the Eylea eyes on every other month Eylea met this criteria of 5 letters, 75 microns? About a third of them did. So that tells you what we already know about at least a third of the eyes, if not more, require more than every eight
Yeah.
week injection.
I'm a huge fan of monthly Eylea. The aflibercept is a great drug. I like to give it monthly, and it's been great
Yep.
now. The HD and the
Yeah.
all the
Yep.
others, all of the other things in the landscape. But this is a great trial, Jay.
Yep.
Congratulations. it's
It,
done really, really well. And so you're reading out in June and in September, maybe.
I would say realistically, July, August-ish.
Right.
You know,
Okay. I
we
mean,
need to the
announcing.
last patient out. The last patient in the first trial was the end of May.
Hmm.
So last patient out end of June, but we've got to, secure the data and make sure it's
Right. It's a
correct.
couple months.
Yeah, it takes
Can
some
I
time.
ask you a logistical question for
Sure.
compliance with a public company? Once you have your results for the first phase three, you have to announce them, correct? You don't wait until the
So
second
the
phase
the
three. I
rule
just don't
is
know. Yeah,
once management knows the result, if it's a meaningful result for stockholders, you have to announce the result within 5 days.
right.
Now, a company can make an exception if the results like don't make sense or something's off. You need to go back and check the data. But basically, there are people in the company that have to monitor the study for safety and other reasons. But once the executives know, you got 5 days.
Right. And let me ask you this. What's your primary endpoint? What are your secondary endpoints? In other words, are you asking
Very
for,
simple.
yeah,
Yeah, very simple. It's visual acuity, visual acuity, visual acuity, and it's non-inferior change in visual acuity from the Eylea control. Now, so we know how the Eylea control is going to behave. Just go back and look at the Eylea control in the last three, studies that have been done. High dose
lots of
Eylea.
supplemental information. In fact, it's amazing that you had to have a control group.
Yes, interesting thought, isn't it?
Yeah.
But we did, and we do, and the control group should gain around 6 letters. That's what we've seen lately in these trials.
Q2
And so
months.
that's what we would expect. And remember, we did enroll 75% naive, 25% previously treated. So the previously treated won't gain quite as much vision, but we'll be in that ballpark. So all we need to do is be non-inferior. Non-inferior doesn't mean the same because remember, high dose Eylea, you remember how it's difference between high dose
Ex
Eylea and 2 milligram Eylea?
Explain it to us.
1.4 letters worse. So you can be non-inferior, but be numerically worse. And that's fine because most of us, and maybe I'm not going to put words in your mouth, but most of us would say, hey, 1 letter, 2 letters, my patients aren't going to notice. I'm not going to notice. So that's okay. So we need to be non-inferior. We need to have continued safety. And this is another big point. We've dosed over 190 patients with our drug in 3 phase 2s and 1 phase 1. We've had no ocular or systemic SAEs reported, and we've had no safety signals. In the phase 3,
That's a lot, Jade. Let me just interrupt you. Why
it
isn't one of
is.
those studies count as a registrational trial?
Good question. The phase 2 trial, the primary endpoint was month 8. It
Got
wasn't
it.
month 12, number one. Number two is we didn't re-inject.
Got it.
We wanted to see how far
Long it
out
would go.
patients could go. And by the
Understood.
way, with one shot, 50% of the patients made it out a year without supplement. But the result
Understood.
of the phase 2 was we were statistically non-inferior, and it wasn't even close, meaning the change between our drug and Eylea was less than half a letter.
Can I
We were,
ask you one other question?
yep.
I have a world of respect, and I know you do too, for Regeneron, Len, and
Mhm.
George. They're amazing company, amazing drugs, amazing
Yep.
science, and they've made a huge impact and great people. And that HD study, the numbers you quoted, there's some explanations to that. We don't have to go into them now, but
Mhm.
and it's a great compound. But do you? it's kind of amazing that you lucked out that you don't have to go up against HD, but
Which
you would have happily, right? Is what you're
it's
saying.
not luck, Steve.
Yeah.
So if you want, and I've learned a lot in this job in a
Yeah,
short
explain
period
it
of
to
time,
us because
which
it's
is one of the
fascinating.
reasons I really like doing it. Is at my age, I'm still learning a lot. It's great,
Hmm.
but a non-inferior trial is based on what's called the non-inferiority margin. The
Hmm.
non-inferiority margin is based on how good is your drug by itself. So the last trial that went anti-VEGF against nothing was the Marina trial.
Hmm.
So the Marina trial was monthly Lucentis against monthly sham. And the sham arm, of course, in general, lost vision, less vision than you'd expect. But in the Marina, it gained 6 letters. So then Lucentis went against Eylea non-inferiority, and Eylea was non-inferior, 2 milligrams. So those are the two drugs that the FDA says if you're doing non-inferior, you have to go against those drugs.
Right.
The other way of looking at it is, let's suppose I have drug A and it's non-inferior to drug B. I use drug B against drug C, non-inferior, but drug C is 1 letter worse. Then I do drug C against drug D. Drug D is 1 letter worse, non-inferior.
Yeah,
You
that's
can see
non-inferiority creep, right?
it's not commutative. That's
Right.
that's the simple logic why you
Right.
can't go against a drug that's
I was
been.
thinking more in terms of the durability. You've got
I'd
durability
love
in your
to,
name.
but Steve, if we're approved, the first post-approval trial we're going to run is our drug against Vabysmo and high-dose Eylea. But
Right.
we're not going to do it for vision. What we'll do it is time to first supplement.
Right.
So if you look at our data, we're going to win against that easily if you look at our
Right.
phase 2 data, if it holds through phase 3.
Right.
And so, again, the big difference here, and this is another important concept to think about these sustained release, especially TKIs, which is a different MOA. And I'm going to digress for a minute because I didn't get a chance to say we have a third MOA that's really important. We
Which
block
is
IL-6.
right.
Vorolanib blocks IL-6 because we block the JAK1 receptor.
And what does
Now,
that do? Explain that to those
well,
of us who
there's
don't know what
pretty
that
good
does.
evidence right now that IL-6 plays a big role in diabetic macular edema. And you may be aware, and your audience may be aware, Genentech ran a phase 2 trial using a monthly anti-IL-6 antibody with Lucentis, and that showed that the visual results were better.
And did they bring it
So,
forward?
oh no, they're not bringing vamikibart forward, probably for two reasons. And again, I'm not, I don't know this for sure, but I think they had a little inflammation problem with vamikibart. But I also think that they probably don't believe that all of us are going to use another monthly injection. So we got very similar results to them in our phase 2, but we did it with one injection every 6 months. They did it with 6 injections in 6 months. So if you said to you of a patient, like you could say to the patient, look, I can give you 2 milligram Eylea and I can get you, 6, 7 letters in 6 months, but it'll take 5 or 6 shots. Or I could get you 6 to 7 letters by week 8, but it only take 2 shots. Which do you want? That's what our phase 2 data showed. We got better vision than Eylea in our Verona phase 2 DME trial at week 4. The curve's already separated.
Yeah, I can
We
tell you
think
what
that's
my
the
patients
IL-6.
I
We
can
think,
tell you what my patients say.
yeah.
They say, "What do you think I should do?"
Right. Well, hopefully we'll convince you. But
Right.
back to how it's going to get used. Every anti-VEGF that got approved after Macugen all said the same thing. Don't use the old one, use us, we're better. Have you ever in your life alternated Lucentis and Eylea?
Only when I didn't have one in the office.
Okay, but you don't do it for, but
Volitionally.
I do believe that two MOAs is likely to be better than one. And I do think that doctors may choose to use our drug because it's long acting, anti IL-6, anti PGGF, and gives you at least 6 month dosing. But on top of it, we'll use a ligand blocker because of the two MOAs. So we're not an either or. We're not saying abandon Vabysmo and Eylea, which is, you said yourself, they're great drugs. We want to be used with them. Now, if our phase 2 data holds, it looks like we could probably take 2/3 of the wet AMD population out every 6 months with our drug alone. But that may not be how doctors choose to use it.
Right.
And I don't think there's anything wrong with two MOAs.
So
Look
let's
at the
just
way
let's
we.
just let's just talk about that for a second because you're hinting at
Yep.
first line therapy.
No,
No.
not really, because remember our phase 3 trial, I already said, you're loaded with Eylea. So we don't know what our label might look like, but one could speculate that label will look something like previously treated or naive wet AMD every 6 months after 3 injections of an anti-VEGF.
Okay, now I'm going to ask you an unfair question, which we can cut out if you want to. I'm sure Big Pharma's knocking your door down to take you over. What's going on? What are you saying to them?
Well, he was saying to everybody, we can, we believe that we can commercialize this drug in the United States ourselves.
Of course you
And we
can. You
think in
commercialize
doing,
YUTIQ, you can commercialize this.
yes, and you know what? It's you can cover all of the US with 50, 60, 70 reps. And a lot of the doctors are concentrated into these big groups. Yes, we're up against two very well entrenched in very, very good companies, but I think we can do this ourselves, which would
What
be
do you
great.
mean which two companies do you think will
Oh,
win?
Regeneron and Genentech.
Right.
But
And
this would be what when you're the CEO of a publicly traded company, you've got you've got really three groups. You're more than three, but two main groups that you really need to please. The patients, got to think about them, and the investors. I'm publicly traded and you bring value to my investors, right? We got the FDA, we've got commercial payers, we've got the doctors, all of those are important. But my point is that when you asked about strategics, my answer is, you know, we think we can commercialize this in the United States ourselves successfully. We probably will be looking for a global partner ex-US, but we're not looking at this point because we believe our company right now is way undervalued.
okay,
Uh,
explain something to me because I don't know about it and I want to understand it. There's another TKI company out there,
mhm,
and it's run by a dear friend of
mhm.
ours and a great doc. And I don't know what the difference is between the two, and I'm sure I'm going to ask him the same question at some
Yeah.
point. But tell me, be
Uh,
politic here, diplomatic, of
yeah.
course, but what's the
I
difference and when are
yeah.
they reading out and do
Yeah.
they have a different MOA and what's
Yeah.
going on there?
So there are a lot of differences, but at the top, I
High
think if you
level.
look at our program, our phase three program, you would say this looks familiar. Non-inferiority, two identical studies, reading out within a couple months of each other. I've read this book before. I've seen it. It works. Ocular Therapeutics has taken what I would describe as a more risky approach to their regulatory pathway for approval. They're doing two very disparate studies. One of them is reading out, we think, probably or in January, so in a month. And that's a Sol 1 trial, the superiority trial. And the other trial's reading out at least a year and a half behind that. So
And is it
it's a
your
different approach.
understanding,
What I
unfair to
would
ask
say though
you,
is again at the
they need
high
both
level.
for registration?
What's that?
They need both for registration as far as you know?
We shall see. I believe that they
That was
likely
an
will.
FDA question, because the FDA has been waving this
Yes,
possible one trial business around.
but Steve, there are actually FDA guidelines that were revamped in 2023 on what you need to do to have a single trial approved, and they haven't changed those guidelines yet. They may be based on Dr. McCary's comments, but they haven't changed yet. So one can look at those guidelines and get an idea of what a company needs to
I
do
have to
to be.
say, regarding Dr. McCary, I saw him speak and met him in person not too
Yep,
long ago,
yep.
and went in with a giant dose of skepticism, and I came
No,
out
he's
super
saying all the
impressed.
right things.
He
It's a
is
matter of
amazing.
we talked earlier about changing culture.
Yeah,
And I have to say the ophthalmic division has been great to work with. They really have. And
unbelievable. The whole FDA has.
we've seen no slowdown. They're superior. But my point again is the FDA publishes guidelines and they can be changed and amended, and they haven't been amended yet.
So
And
let me
therefore,
ask you this. I'm
Mhm.
sorry for interrupting, but Ocular Therapeutics and your company, MOA, similar, different?
Similar, but they don't block IL-6 and we do. And their drug delivery system is a hydrogel and ours is different. Uh, but in saying that, the other thing your listeners should understand is if you are doing a single trial for approval, your p value is no longer p less than 0.05. It's P less than 0.05 times 0.05. So you need a really, really high low p-value to hit if you're doing single trial
Right,
to
right.
make it statistically relevant.
Well,
And
it's
there's
super
other
exciting.
things considered. The FDA in their draft guidelines said you have to have 400 patients for safety. One trial, the way it's designed currently, is not going to give you that. So, we'll see. I do want to say at the high level, though, there's room for two TKI companies. This is a huge market. It's 10 billion market in wet AMD and growing. And we're the only TKI going into DME. That's a 3 billion market. And we've got that IL-6. So I really like our positioning. What I've also learned from very experienced commercial folks is when you're introducing a new MOA to a group, whether it's retina specialists or oncologists or whatever, it's always better to have two companies doing it because it's easier to understand and easier to accept. So we wish them luck. We're tied at the hip. I like our program. I wouldn't change a thing about it. And I would say overall, I think EyePoint has taken a more de-risked approach.
I think it's great for you that they're reading out first, and I think it's great for you that they're going to do a great job. They've got a great team,
Yep,
great investigators, great leadership, and rising tide floats all boats. You're exactly right. It
absolutely.
is 100%. It's in that movie Arthur, I think. Yeah, I like a living room that's big enough to park an airplane in.
Yeah.
It's a
And you know
big
what?
space.
Retina's never been a zero-sum game,
Right,
ever.
right.
Look at how it's expanded and expanded and expanded. This is not a zero sum. There's room for both. I do think though, again, if one or both of us are approved, I think that it will be again difficult to think about other ways of getting sustained release that would be successful. What I'm getting at is, you know, a surgical sustained release for wet AMD, we can argue how viable it is to a big population. But if you have an injectable that's bioerodible and can be repeated by the doctor if they choose to or not choose to, that takes away some of the permanence of things like surgery. And so that's another thing to think about should one or both of us get approved. And it's really a two-horse race right now, as you may be aware. Clearside also was in the TKI space with a suprachoroidal injection, but unfortunately, we're unable to find funding for their phase three.
Right. Well, another good company with great people in it, and it's
Absolutely. There's still there that injector is obviously approved and valid, and that's not going away. Uh,
yeah. So, what's your biggest challenge been being the CEO of a public company? New challenge. I mean, I know you know the retina community inside and out, the basic
yeah,
science, the MOA, super exciting, well-designed trials, execution's been great.
yeah.
Everybody looks to you as a bellwether for excellence. But personally, what's the biggest challenge of being a public CEO?
So for me at the beginning was uh understanding how good a team I have. Meaning, what happens in all of these companies is that you have a plan. And then, like Mike Tyson, you get punched in the face and you say, okay, I got to change the plan. And you have to be able to do that. And when you learn that you have great people around you that are hardworking and smart, you suddenly become a lot less worried and a lot fewer sleepless nights because things will come up and we will brainstorm it and we'll work together as a team and we'll listen to everybody and get the right idea. And the problems, we've solved them all. We've solved them all. So I'm sleeping a lot better now than I used to. Uh, but I would say the general comment here would be the financial aspects of things. I really knew very little about public companies, the rules around public companies, the way they raise funds. Uh, and, I think at the beginning, I think some of the investors were skeptical that I would hang around. They were, well, you've been an academic all your life. As soon as things turn south, you're going to quit. I kind of heard that. And obviously, it hasn't happened and it's not going to happen. But I have a really good CFO, George Elston. I had good mentors here. I've got a great board, really experienced, and they've really helped me
Who's
learn.
on your board?
Joran Ando is the
Mhm.
chair of our board. Joran's been in ophthalmology before. I think he was the CEO of Pharmacia years ago. Wendy DeChico, head of our audit committee, Stuart Duty, a former Guggenheim banker, Fred Hassan. Fred's been the CEO of multiple companies, including ophthalmology companies. Fred is just
Amazing.
yeah, this is my analogy for Fred. It's like if you show me an OCT, I can tell you in 5 seconds what the disease is. It's pattern recognition. I have a problem running the company. I can call Fred and say in 5 seconds, he'll say, well, this is what you should do. He's
Right.
been there, he's been done that. It's great.
It's pattern
John,
recognition for him.
yeah, John Landis, who's an experienced CMC guy. Karen Zatteridge, again, old. She was a CEO, very successful publicly traded company. And of course, Nancy Lurker, who was the former CEO. And finally, last but not least, a name that most of us know, Reggie Sanders.
Oh, look
Uh,
at that. I didn't know Reg
yeah,
joined your
Reggie
board.
joined our board about a year ago, and he's been a
Oh,
great addition. The thing is, when you're the only retina specialist in a retina company, it's pretty easy to be in an echo chamber.
yeah.
And Ramiro Ribeiro, who is now our chief medical officer, people may know him from Apellis. He's a retina specialist. Anna Dimitriadis joined our clinical team. She's not a retina specialist, but she's an ophthalmologist. So we're not really, they don't look to me anymore and say, well, Jay's word is the word because he's the only retina specialist.
Well,
What's
that's
great
really
about
good
having,
because you don't want to be the problem solver. You
no,
want to be the problem solver of very last resort. But I will
correct.
say this, I did not realize that Reg joined your board.
Yeah, and it's been
I've been
great
on a
because
board
the
with Reg.
board looks
I,
to Reg and say,
I've been
Reg,
on the board with Reg, and Reg
yeah,
he's brilliant, he's incisive, he doesn't need to hear the sound of his own voice.
yeah.
He is a great board member, and he
You
has an
summarized it well. And
infectious
when I
joy,
first I first asked him, he was like, Jay, I looked at your board members. I don't resemble any of them. Why do you want me on the board? I
That's
said,
the
Reg,
reason.
that's exactly why I want you on the
Yeah.
board. You have a background that's unique. I mean, Reg essentially invented the business of retina. He's got you summarized him well. He has got great insight, and that's exactly what you want on a board. You don't want cookie cutter. You don't want people who's from the exact same background. And
And by
because
the
you need
way,
strategy.
his experience coming up the way he came up and building what he built, I've got a lot of former fellows in that group. He
Yeah.
is a great leader. And
Yep.
he's got his fingers in the business of medicine all over the place.
And
We were on the SRS board together and he was
we were
fabulous.
residents together at Will's
Oh,
way back when.
there you go. There you
Yep.
go.
We meet
There you go.
again.
Jay Duker, thank you so much for sharing all of this with us. And I'm sure that our listeners are going to be reaching out to you and looking for you and finding you on all those publicly available emails and phone numbers and coming up to you and thanking you for sharing these
Yep.
insights. It's been really exciting and informative. I've learned a lot, and I wish you nothing but good luck and a happy Hanukkah, Merry Christmas, and all the other festivals of joy and happiness that you celebrate. And congratulations on this career trajectory and your newest journey. And I know it's all consuming at this point, but it's we're really looking to you for major benefits for our patients and for us as physicians. So thanks, Jay. Really appreciate
Thank
it.
you, Steve. It's been a lot of fun.
If you enjoyed this video, please visit us at RetinaRounds.com. There you can sign up for our email list. You'll get a notification every time a new video is posted. And if you have an interesting video or a tip or trick that you'd like to share, please follow the links on our website and you can upload your video there. Thanks so much for watching.